structure of proteins. So by the end of the lesson, you should be able to draw the possible primary, secondary and tertiary structures of a protein. You should be able to explain how a quaternary structure is formed and give an example. And you should be able to apply the knowledge to show how a protein shape is related to its function. So a polypeptide is multiple amino acids joined together by a condensation reaction. So lots more condensation reactions occur to form a polypeptide. So you get many peptide bonds, and remember, with condensation you get water molecules that are formed or released from the reaction. So the primary protein structure is literally the sequence of amino acids in the polypeptide chain. So you could have alanine, glycine, leucine, glycine. It just depends on what order those amino acids are in the chain. So it's the sequence of the amino acids in the polypeptide chain. The secondary structure is where the polypeptide chain either coils into alpha helices, which you can see here, or it folds into beta pleated sheets. And the secondary structure, the alpha helices or the beta strands, are held together by hydrogen bonds between the amino acids. So you can see here this alpha helix has hydrogen bonds. These little squiggles here. The hydrogen bonds hold the secondary structure together. So, tertiary structure. So once that alpha helices or beta pleated sheets have been formed, they then fold, the secondary structure then folds on itself again and again, twists and folds, and it is held together by different chemical bonds, giving the protein a complex 3D shape or tertiary shape. And the bonds that are holding it together are disulfide bridges between the sulfurs of different amino acids. They're very strong bonds, so the disulfide bridges are very strong. You've got ionic bonds, which are very strong, and they are between the positive and negative charges of different amino acids. And then you've got weak hydrogen bonds. So the three bonds that hold the tertiary structure together are disulfide bridges, ionic bonds, and hydrogen bonds. And what you can sometimes get is a quaternary structure of a protein. And this is either more than one polypeptide chains joined together, or sometimes the polypeptide chains can be joined to non-protein groups called prosthetic groups. And these can combine with the polypeptides to form a quaternary structure. And a good example of this is a hemoglobin molecule. So you can see here you've got 4 polypeptide chains, 4 separate ones. You've got 2 alpha subunits, 2 beta subunits, but then associated with these polypeptide chains, you've got these prosthetic groups, these non-protein groups called heme groups, which are ion groups, and that is where the oxygen will bind to carry oxygen around the body. Another example of a quaternary structure is collagen. So collagen is a fibrous protein consisting of 3 polypeptide chains wound together, round around each other, a bit like on a rope, where the fibres are wound around each other. And each of the 3 chains is a coil itself. So it's coiled and then coiled around each other. And hydrogen bonds form between these coils, which are around 1,000 amino acids in length. And it's these hydrogen bonds that give the collagen its structure and its strength. So, looking briefly again at the functions of proteins, we did this in a previous lesson, but you have fibrous proteins, which are structural proteins like collagen. You have globular proteins, which have a metabolic function like hemoglobin or enzymes, and it is their structure that allows them to carry out their functions. And the 3D structure, the tertiary structure, is very, very important in carrying out its function. And it makes the proteins distinctive in interacting with other molecules. But ultimately, the tertiary structure is determined by the primary structure, those amino acids, because they will determine where the bonds form. So let's do a few practice questions. So first one, what are the monomers of proteins called? So the monomers, individual units, are called amino acids. What is a polypeptide? It is a chain of more than 2 amino acids joined together via condensation reactions. Draw the general structure of an amino acid. So you've got the central carbon bound to a H, bonded to an R, the variable group, the R group. Then you've got a carboxyl group, C double bond O, OH. You've got an amine group, which is NHH. And that is your amino acid structure. What sort of reaction links amino acids together? Be a condensation reaction. What is the name of the bond that forms between amino acids? It is a peptide bond. Name 3 bonds that may be formed between the amino acids in a polypeptide chain to form the tertiary structure of a protein. So you've got hydrogen bonds, ionic bonds, and disulfide bridges. Explain how the shape of structural proteins make them specialised for their function. So structural proteins are made of long polypeptide chains lying parallel to each other with crosslinks between them. This makes them physically strong. The Biret test is used to test for proteins. What is added to the test solution to make it alkaline? It is sodium hydroxide solution. What is added next to the solution? It is copper 2 sulfate solution. What would a positive test result look like? It would be purple or violet.